Hi, everyone. Welcome back to the 2025-2026 Oncology Pharmacy Residency Clinical Pro Series. Today, I'll be presenting on the CARTITUDE 4 trial and the new follow-up results that were released this year and compared them to the original 2023 results. For this presentation, I will go over the background of the CARTITUDE 4 trial, describe its parameters, strengths, and limitations of this clinical trial, and then describe the implications of the results of this study. To start with the background, ciltacabtagene autoleucel, which I'll be calling cilta-cel for the rest of this presentation, was approved by the FDA on February 28, 2022 for the treatment of adult patients with relapsed refractory multiple myeloma after 4 or more lines of prior therapy that included a proteasome inhibitor, an immunomodulatory agent, and anti-CD38 monoclonal antibody. This CAR-T therapy was approved based on the results of the CARTITUDE 1 trial, in which 97 patients received the phase 2 dose of 0.75 times 10 to the 6 CAR-positive cells per kilogram. And they found pretty good efficacy results where the overall response was 97%, the 1-year progression-free survival was 77%, 1-year overall survival rate was 89%, the stringent complete response rate was 67%, and then the time to first response was 1 month, while the median duration response has not been reached. There are other standard of care treatments that can also be used for treating relapsed refractory multiple myeloma, in which the NCCN guidelines does have a category 1 recommendation for daratumumab plus pomalidomide plus dexamethasone. And this is based on 2 clinical studies in which the MM014 trial is a phase 2 multicenter multi-cohort open label trial in which 112 adult patients were given this regimen, and they found that after a median follow-up of 41.9 months that their median overall survival was 56.7 months, and then the overall response rate for these patients was 78.6% overall, in which patients who relapsed on lenalidomide had an overall response rate of 81.5%, while patients that were refractory to lenalidomide had an overall response rate of 77.6%. There was then a comparison in the Apollo trial between the daratumumab plus pomalidomide plus dexamethasone arm versus the pomalidomide plus dexamethasone arm. And they found that there were better efficacy results with the addition of daratumumab, in which the median progression-free survival and overall survival were both longer, but it was not statistically significant for the overall survival results. Another recommended treatment regimen for relapsed refractory multiple myeloma, that is a category 1 recommended treatment option, is pomalidomide plus bortezomib plus dexamethasone. This was evaluated in the Optimism trial that was conducted at 133 hospitals and research centers in 21 countries. And this compared bortezomib plus dexamethasone with the addition of pomalidomide. And it found that there was a statistically significant difference in median progression-free survival when pomalidomide is added to dexamethasone plus bortezomib. Shown here are other treatment options that are recommended by NCCN for relapsed refractory multiple myeloma, where you can see different treatment regimens depending on what the patient was refractory to. I have also highlighted the 2 treatment regimens that I previously discussed for lenalidomide refractory multiple myeloma, since this is the patient population that the CARTITUDE 4 trial specifically enrolls. For the study objectives, they then compared cilta-cel with these 2 standard of care treatments that was shown on the previous slide. For this clinical trial, it is an open-label, multi-center randomized phase 3 trial in which Johnson Johnson was the sponsor of this trial and is also the manufacturer of Cilta-cel. For patients that were included in enrollment, they were adult patients with multiple myeloma who had 1 to 3 prior lines of therapy, had prior exposure to a proteasome inhibitor and immunomodulatory drug, and has had documented evidence of disease progression on or within 6 months of their last treatment regimen. Patients were excluded if they've had any prior CAR-T therapy or anti-BCMA therapies, and then they were also excluded if they had any autologous transplant at least 12 weeks before apheresis or allogeneic transplant less than 6 months before apheresis, and also if they've had any health condition that would predispose them to unnecessary harm or confound efficacy outcomes. For the interventions, they compared Cilta-cel with the 2 standard of care treatment options that were previously mentioned in the background information. It is important to note that in this trial, the majority of patients in the standard of care arm did get the daratumumab combination treatment, where it was 183 patients, while the remainder got the bortezomib combination therapy. They then looked to evaluate the extent of efficacy outcome improvement with the outcome measures shown on this slide. For statistical analysis, they use standard statistical measures such as the Kaplan-Meier estimates to analyze 30-month rates of treatment-free survival, overall survival, and progression-free survival. They also determined that the study population of 400 patients and 250 progression-free survival events would achieve 90% power to detect a progression-free survival hazard ratio of 0.65 with an overall two-sided alpha level of 0.05. The two-sided p-value that was required to establish superiority of Cilta-cel over standard of care for progression-free survival was 0.0191, where this threshold was based on 187 progression-free survival events occurring. For the patients that were enrolled in both groups, the baseline characteristics were pretty similar. And what's notable is that there was a majority of patients in both groups that had high-risk cytogenetics, and they also did have a similar percentage of patients that were refractory to triple-class treatment as well. The efficacy results for Cilta-cel in this newly released trial results continue to show better efficacy outcomes for Cilta-cel, in which the 30-month progression-free survival rate remains higher in the Cilta-cel group, and the overall survival rate also remains higher, in which the 30-month overall survival rate was 76.4% in the Cilta-cel group compared to 63.8% in the standard care group. And this is also notable because in the previous 12-month overall survival rate, it was found to not be statistically significant, and the overall survival rate was pretty similar in that 1-year survival rate percentage. For other efficacy results, the median progression-free survival was not reached in the Cilta-cel group, while the standard of care was 11.8. The median overall survival was also not reached in both groups, but it was found to be statistically significant, in which Cilta-cel was favored. Overall response was also higher in the Cilta-cel group. Complete response or better was higher. MRD was higher in the Cilta-cel group as well. And then the median time to worsening was not reached in the Cilta-cel group, while standard care was 34.3 months. But what's notable is that there was many patients that were censored for this time to symptom worsening, which is one of the limitations of this clinical trial. For the safety results, both treatment arms had all patients who experienced drug-related adverse events, where a majority of them did experience at least grade 3 drug-related adverse events. There was a slightly higher incidence of grade 5 events in the cell-to-cell group. For the most common adverse event that they did experience, it was CRS in the cell-to-cell group, which is to be expected, and infections for the standard of care arm. Ultimately, the investigators did conclude that cell-to-cell would significantly improve survival outcomes and quality of life in patients with relapsed refractory multiple myeloma compared to standard of care treatment. And this clinical trial did have many strengths where the majority of patients that were examined had higher risk cytogenetics, so it would give us a good idea of how efficacious this could be for patients that have worse prognosis. The efficacy outcomes were also assessed in an intention to treat population, which is representative of real life practice. And they also did have similar baseline characteristics and were mostly representative of patients that have multiple myeloma in the United States. Another really important study strength is that they also did compare cell-to-cell with other standard of care treatments that have been found in clinical trials to be very effective as well. So this would allow clinicians to see which treatment would be best for a patient population that has not had success with other standard of care treatment options. Despite the strengths of the CARTITUDE-4 trial, there were a number of study limitations. One notable limitation is that the standard of care treatment arm used different bortezomib dosing than what is currently recommended by NCCN based on the Optimism trial. The dosing that was used for the standard of care arm was 1.3 milligram per meter square, twice a week for 2 weeks for 8 cycles, then once a week for 2 weeks per cycle. When it came to the patient population, there were a number of exclusions to note. One was that patients of African descent were underrepresented, and this patient population has the highest incidence of multiple myeloma in the United States. Patients with any prior anti-BCMA therapies were excluded as well. This would have been helpful to include because patients who have been on therapies such as teclistamab or lemvositamab could potentially see benefit that is not known due to this exclusion. There were a number of patients in both treatment arms who were not evaluable for MRD due to lack of post-baseline samples for disease progression or subsequent therapy. Lastly, a majority of patients in both treatment arms were censored from the time to symptom worsening questionnaire, and this would have been helpful to see because it would have painted a more accurate picture of how much the quality of life could improve with these treatment options. Ultimately, it is important for us to consider the follow-up results that were released this year for the CARTITUDE-4 trial because the NCCN guidelines do list cell-to-cell as a preferred category 1 treatment option for patients with lenalidomide refractory relapsed multiple myeloma after one prior line of therapy that includes an immunomodulator and a proteasome inhibitor. This is reasonable for NCCN to recommend at the time of this recording because the 30-month overall survival rate was higher in the cell-to-cell group with a bigger difference than what was seen in the original 2023 trial result release with the 12-month overall survival rate that was statistically insignificant and had a lower difference in these rates. This is only expected to continue to show a bigger difference as more longer-term follow-up results are released. And then this is just to show where cell-to-cell is in the guidelines at this time of the recording. Here's my references. If you do have any questions about the contents of this presentation, please feel free to reach out to me at cchen1@mindmed.health. Thank you.